Okay, welcome to the review session for Exam 2. Since I don't have any questions, I'm just going to go over a few items that came up in the exit survey to clarify that information. And I'm going to start by sharing the screen with you so that you can see the study guide. So I just want to remind you to focus on the topics that are listed in the study guide, especially these open response topics. You should be writing all of the information in your notes about these topics. Just to clarify, you should focus on the function of the bacterial cell wall in the environment and how it protects the bacteria and maintains the integrity and the viability of the bacteria. Also, you should know the key differences between the gram-positive and gram-negative bacterial cell wall and how those differences will account for differences in infection. Or different susceptibilities to treatment. And then you should also be able to compare the structure and know all of the internal kind of anatomy of the bacteria versus eukaryotic microorganisms like fungi. So that's just an overview. Be sure that you're writing all your notes and practicing writing that information kind of from memory before you come into the exam this week. So some of the questions that came up in the the exit survey this week included there was still some confusion about osmosis as well as the difference between the cell membrane and the cell wall. And these are really key concepts to know. The cell wall is very different from the cell membrane in its structure and in its function. So let's take a moment to look at that. And that is in chapter 5. Okay. So chapter 5 material goes over these the composition of the bacterial cell wall, both in the gram-negative, which is shown here at the top, and the gram-positive. And remember that there are key similarities, and there are also key differences in the structure of the cell wall. So these cylindrical, alternating colored, kind of striped cylinders represent the alternating glycan or sugars in peptidoglycan, and that's present in a very thin layer in the gram-negative, where you have many, many layers, up to 40 layers of peptidoglycan in the gram-positive cells. In addition, there's no outer membrane present in the gram-positives, but there is in the gram-negatives. So what does this mean? The cell wall, as you can see, has a very different structure than the plasma or cell membrane, or even the outer membrane in the case of gram-negatives. So membranes, by definition, are biological membranes are two layers, a bilayer of phospholipids. So the major macromolecule in that structure are lipids, specifically phospholipids. So you can see that's the yellow balls with the gray kind of tails of the lipid portion, the balls representing the phosphate group that joins the two lipid tails. So, and they're two layers, whether you're looking at the cell membrane of gram-positives or gram-negatives. So how is that different than the structure of the cell wall? Well, as you can see, the cell wall is these many, many layers of glycans, repeating the two glycans, the N-acetyl glutamic acid and the N-acetylmuramic acid. So nag, nam, nag, nam, nag, nam. And they form this kind of lattice. You can see it's just not as densely packed as the membrane. And so just inherently by that structural difference, that should help you to remember that the membrane is selectively permeable. It doesn't let everything that's outside the cell in or that's inside the cell out. So that's the important functional difference between a membrane, whether it's the inner membrane or the outer membrane, and a cell wall. Cell walls let everything, that everything of various size, charge, whatever, through all those multiple layers that you see here. But then they get stuck at the cell membrane, and that's what determines what actually gets into the bacteria. As you can see here in the gram-negative, there's a membrane on the outside of the cell wall. So even before that, those glycan layers or that single layer of glycans, there's a selectively permeable membrane that's preventing most of the things out in the environment from getting in. And that's where these specialized proteins that are only found in gram-negatives called porins, which is the line labeled C is pointing to. They're these transmembrane proteins that allow only select items to get through, proteins or sugars. But there are many different kinds of porin proteins, and each one has a specific shape and size, so it will exclude most molecules and only allow a select few to get through, few types. So that's one of the main differences I want you to remember between the membrane, cell membrane, and the cell wall. Now the cell wall is, especially when it has many layers like in the gram-positive, is what gives the strength and determines the shape of the cell. And that strength can be broken down in the case of treatment with antibiotics that target the cell wall. That will weaken the cell wall such that the bacteria become susceptible to changes in the environment, in the tonicity of the environment. Okay, so that brings me to the next topic that you really should know for the exam, and that's how bacteria live in these environments that are not isotonic. So remember, bacteria live pretty much they're ubiquitous, and so we can find them anywhere in almost any environment on Earth. And because of that, they have to be able to persist and actually grow and thrive in environments like the ocean, where it's a very high amount of solutes outside of the cell, so it's a hypertonic environment, or in freshwater. Most environments are hypotonic, very little solutes outside of the cell, very dilute. So in that case, and let's look at this figure from chapter 4. So normally if you're looking at a cell that doesn't have a cell wall, you can see what would happen in like a saltwater environment like the ocean is that the cell would just shrivel up, and that has to do with the movement of water. And the water is trying to move out of the cell so that the concentration of these solutes represented with the red balls is pretty much equal inside and outside of that membrane. In order to survive, bacteria that some bacteria this will happen, and they'll undergo what's called plasmolysis, where the cell wall doesn't change, it maintains the shape of the cell, but the cell membrane will shrivel in a hypertonic environment. But other bacteria are able to adapt and actually live in the ocean as their habitat. So why can they do that? They produce extra solutes inside the cell, other ions like potassium rather than sodium, which would throw off the function of the enzymes and other parts of the metabolism of the cell. But they'll make extra potassium, for example, in order to bind up the water so that it's busy kind of stuck around these ions that's present inside the cell, and so the water is no longer free or available to move across the membrane by diffusion to compensate for that difference. And so it fools the bacteria and basically fool or trick the conditions tip them in their favor, so that there's no longer a shriveling of the cell wall because there's no movement, or very, very slow movement of the water out to the outside of the cell. In the case of bacteria that are in hypotonic environment, if it wasn't a bacterial cell, if it had no cell wall, then eventually, because of the movement of the water into the cell, you would get the swelling of the cell to the point where it would burst and the bacteria would die. Really what the cell wall does is it doesn't shift like the cell membrane. It's not as flexible or fluid. Remember, sometimes the membrane is described as fluid mosaic. There's the fluid mosaic model for the cell membrane. That's not true of the cell wall. It's fixed. It's covalently linked by these cross bridges in between the many layers. Let's go back to here. So those are represented on this diagram in C as the dots that are the amino acids in these short proteins that covalently link the many layers to each other, as well as the alternating glycans in a single layer to each other. So it's really strongly bound together, covalent bonds being the strongest bonds. And because of that, even though there's an influx of water into the bacterium, the cell wall prevents the enlargement of the cell. It keeps it fixed at its size. And so it resists this turgor pressure that builds up against the cell membrane. And the bacteria can survive and function within those environments. So bottom line, you can think of the cell wall as kind of the skeleton that really holds the structure. You know, without a skeleton, our bodies would just be kind of like a pile of tissue, kind of like Jabba the Hutt or something from Star Wars. But for the bacteria, they have this cell wall that's really essentially a skeleton that's holding their shape, determining the shape, maintaining the shape of the cell. And the membrane doesn't do that. It's flexible. It responds to these changes in tonicity in the environment. But it's an important barrier that determines what gets into the cell and what gets out of the cell. Okay, so I've gone over that question. So remember, just as far as the selective permeability, so what gets in by simple diffusion is only uncharged non-polar molecules. Water can diffuse across even though it's only slightly polar, and so it can diffuse across. So water is one of those things that can get freely across the membrane either way. Small molecules like gases like CO2 and oxygen will freely cross the membrane. But larger molecules like proteins or charged molecules, which proteins are, or small molecules that are charged like ions like sodium or chloride, potassium, they need special transporters. Proteins depicted here in this diagram in purple that go all the way across the membrane. So they're called transmembrane proteins. They're channels, or that can be selective based on charge, or protein transporters that are carriers of molecules, and they only carry one type of molecule because of specific interactions between that molecule and the transporter. So for example, glucose, too big to get across the membrane by diffusion, so it has to get in by a transport protein. Now sometimes it is transported in at the same time. Often in bacteria it's transported in at the same time as ions like sodium ions. And that way it doesn't require an input of energy. But other times, so in that way it's facilitated. Facilitated can also be diffusion of, movement of solutes such as glucose down its gradient. So it's sort of like diffusion, but it doesn't happen right across the lipid bilayer. It needs this special protein, so we call it facilitated diffusion. And then in the case of movement of water, there are also special proteins where water moves through, called aquaporins. And that's where the vast majority of the water will be moving through those specialized proteins. But some can also diffuse across the membrane. And that's when we're talking about the movement of water, and water only, not ions or sugars or any other molecules like proteins, then we use the term osmosis. That's just to describe the movement of water in or out of a cell. All right. So let's go back to the study guide. Be sure that you know, remember you're only responsible for part of Chapter 3, just about the different types of stains. We already did gram staining in lab, so review that, review the steps, and the differences between the gram-negative and the gram-positive cells and how they respond to each of those staining steps. Then from Chapter 4, just know the role of the cell membrane, its selective permeability, what I just went over, and osmosis and tonicity. Also be sure that from Chapter 5 there's a lot of information about the prokaryotic cell structure. Be sure you know that the difference between that and the eukaryotic cell structure. What kind of macromolecules, macromolecules are reviewed in Chapter 4. Be sure that you know the 4 major organic macromolecules, so the proteins, nucleic acids, lipids, and carbohydrates that are all necessary and found in all living things, including all microorganisms. But understand the differences. For example, where is DNA stored in a bacterium? It's different site than in a fungal cell like yeast. And then be sure to review the differences between gram-negative bacteria and gram-positive bacteria, so those external structures, the cell envelope and how it differs, and know other features of the cell envelope in bacteria that are external to the cell wall, things like capsule, S layers, things like that. And then don't forget also to, from lab, to review the relative size of microbes. There's a nice figure in chapter 3 about that, and the difference between the morphology, so how we describe what we see for the microscopic morphology. Remember, we looked under the microscope to look at just a single cell, versus the colony morphology. Remember, we were looking at that on the plates that we had touched our cell phone to. So colonies being many, many bacteria, but they have a characteristic, maybe color and size and sheen, that can be described as colony morphology, whereas microscopic morphology don't use those same kind of parameters um, to describe a single cell. Be sure you review the vocabulary, and you can try these questions in your book. You'll find the answers at the end of the book. Okay, so that's all I'm going to go over. If you do have any other questions, don't hesitate to log on to our Moodle website and post them in the Ask the Professor section at the very top of our homepage.